Thank you so much for the introduction, and thank you to the organizers for the opportunity to talk about our work. So as Moose said, I'm a postdoc in the lab of Danny Bassett, and today I'm excited to tell you about some of our work using network control theory to study neural state transitions. So I'd like to start off by just introducing the network control model and how we use it to study neural dynamics. I'll talk about some recent studies from our group that have begun to look at the relationship between cognition and control energy, as well as underlying neurobiology. And then finally, I'll talk a little about some ongoing work that's aimed at extending the network control model to multiplex representations of the brain. So network control theory is a systems engineering approach that determines which perturbations can drive the system to a desired state. It's typically been used to study mechanical systems, air traffic control systems, and robotics. And relatively recently, it's begun to be applied to the study of neural dynamics, which is what I'm going to be focusing on today. So our control model is a linear model of neural dynamics across time. Within this framework, the A represents a weighted adjacency matrix that encodes this brain structural connectivity, such as what you would derive from diffusion tensor imaging of the human brain, for example. XT represents the state of brain across time, and is essentially a vector of neural activity across the whole brain. B is the number of regions being given control input. And then U is the control energy, or the amount of control input required to drive the brain from one state to another. So what are the inputs and outputs of our model? Given some map of the network or the structural connectivity, and some model of dynamics or different neural states that the brain occupies and how the brain moves between those states across time, we can determine how to perturb the system to control its trajectory, and we can compute the amount of control input or energy necessary to drive the brain from one state to another. Okay, so the first input that we need is the A matrix, which encodes structural connectivity. So in humans, this is most often derived from diffusion tensor imaging, as you see here. In species like the mouse or macaque, we might use data derived from track tracing studies. And then in species like C. elegans or Drosophila, which we've seen already some really beautiful examples of in the earlier talks, we could use electron microscopy to derive a structural connectome. Then the second input that we need is a model of neural dynamics. So in other words, we need to know what neural activity states the brain occupies and how it transitions between those states. So within the network control theory framework, we can imagine different brain states, which are represented by distinct patterns of neural activity, as occurring along an energy landscape where further away states are more difficult to reach and require more control input. And these brain states could represent different experiences, different cognitive states, different objects.
Sorry, maybe this will answer it, sorry.
Is this what you were going to ask?
I think so.
Okay, great. So in addition to identifying brain states, that we need a model that explains how we transition between them. And so a challenge here that's already been discussed a little bit in the meeting is to choose a model that accurately reflects neural dynamics but is simple enough to give us readily interpretable results. So here you can see in order of lowest to highest complexity, we have linear time invariant models, followed by linear time varying models, followed by nonlinear models. So while we know that at the cellular level, neural dynamics are nonlinear, recent work from our group has actually shown that linear models perform better at approximating macro-scale neural dynamics like what you would measure using fMRI or EEG. And that seems to be the case because some combination of noise and filtering and averaging over the activity of many neurons within a region is actually masking those nonlinearities that we would see at a smaller scale of resolution. So the model that we use is linear and time invariant. However, if you were thinking about microscale dynamics, it might be more appropriate to consider a nonlinear model. Yeah.
So should I think about the energy you're talking about as being like the Lyapunov function of this linear system, or
As the what?
like Lyapunov function of the linear system, or energy function of the? Is the relationship between the E function and the linear dynamical system that you have?
It's the.
Sorry, how is E? You're talking about energy.
Yes.
How is that defined in terms of the linear dynamical system?
I'm going to get to that in 2 slides. But real quick, if anyone's interested to read more about the performance of linear versus nonlinear models at approximating macro-scale dynamics, you should check out this paper. It might be like 4 slides instead of 2. Okay, so now that we have a map of the brain's connectome and a model of neural dynamics over time, what kind of outputs can we obtain from our control model? One would be the system's response to a control input that drives it along a controlled trajectory away from its natural trajectory. The controllability of the system, or the ability to drive changes in its state with control input, and the control input that's required to drive the system to a desired state. So this last output is the one that I'll be focusing on today, as it's most relevant to thinking about how we might perturb neural dynamics to affect a desired change in state, and to consider which changes may be harder or easier. Okay, hopefully this will get at your question. So in our network control model, we can define a cost function that penalizes control energy and distance from the target state. And this intuitively makes sense, since the energy cost term constrains the range of time-dependent control energy, so the brain cannot use an infinite amount of energy to carry out a task or a function. And the distance constraint reflects the fact that optimal transitions between states should have reasonable lengths. So in practice, the control input could be something external, like stimulation, or it could be something internal, like cognitive control.
Sorry, was your target time dependent? Like you're integrating over time here?
Yeah, so there is a time horizon parameter that we choose.
You're comparing where you are now from a target of where you want to be now.
Exactly.
And you actually have a target all the way along your sort of time horizon.
Yeah.
So is an RG function defined over pairs of states? Like a current and a
Yes.
final?
Yeah, exactly. Okay. So now I want to get into talking about some recent work from our lab that has started to examine the relationship between control energy and cognition and underlying neurobiology. So the first study that I'm going to talk about is set out to test the hypothesis that cognitive states that require more effort to reach and maintain will be associated with greater control input. So specifically, this study examined fMRI from young adults who engaged in one of 2 cognitive strategies as they're reacting to alcohol cues in the scanner. So they were instructed to either just react naturally to the cues, or to react mindfully in such a way that they mentally distance themselves by sort of observing non-judgmentally their response to the cue without getting caught up in the response. And then a control theoretic approach was used to assess whether reacting mindfully to an alcohol cue would require more input than reacting naturally, which would be consistent with the idea that control input reflects cognitive effort. So the first hypothesis was that more control input would be required for the brain to reach the mindful state than the natural react state. And then the second hypothesis was that the mindful state would be less stable than the natural react state. So in other words, the brain would be more likely to transition out of that state. It requires more control input to maintain the neural activity state associated with mindful reacting. So here control input was given to regions of the dorsal attention, ventral attention, and frontoparietal networks, which are associated with cognitive control. And then the brain states in this study are beta weights from a general linear model of the fMRI data. So they reflect task-associated activity. And so what you can see is that the mindful attention state required more control input into these cognitive control regions to reach. So this is a case where we're going from an initial state of 0 to mindful or natural react beta weights. And then also we observed that the mindful react condition was lower in stability, meaning that it was more costly to maintain that neural activity state. The brain is more likely to transition out of that state. So this is consistent with the idea that evoking a cognitive regulation strategy is more effortful, requires more input than just reacting naturally. Okay, further evidence for a relation between control and cognitive efforts comes from work in which we've looked at the relationship between control energy and the development of habitual behavior. So in this study, we used network control theory to model neural state transitions in 2 macaque monkeys that were performing a saccade task. So the monkeys engaged in repeated trials where they would saccade over this grid of 9 points. And once their gaze crossed a baited target, they would be rewarded with a juice reward. So over time, the monkeys' behavior became more efficient and automatic as they figured out the best strategy to identify that targeted or that baited target and earn the reward. During the sessions, multiple electrodes recorded populations of neurons throughout the cortex and caudate nucleus. And these recording channels were treated as nodes in a network. So we recorded average firing rate for each channel and then used that to derive an effective connectivity matrix, which we used as the structural matrix for our control analysis. And then the average firing rate across regions served as brain states associated with each trial. So here we're just looking at average control energy over the course of sessions for each of the 2 monkeys, monkey G and monkey Y. And so we observe generally downward trends over the course of sessions, suggesting that the transitions become less costly as the animals adapt to the saccade task and their behavior becomes more efficient. And then we also defined several behavioral metrics that reflect features of habits. One of these being similarity factor, which represents the similarity between 2 saccade patterns performed one after another in the same session. So this metric can be used to assess whether the monkey is performing increasingly similar patterns the longer she engages in the task. And we observed a negative relationship between control energy and similarity factor, suggesting that performing more similar saccades in sequence, as we would expect to see as habitual behavior is developing, is associated with lower energy cost. Another metric that we examined is cluster label entropy. And this was obtained by performing hierarchical clustering of the different saccade patterns performed over the course of trials. And this metric addresses the question of whether the monkey is choosing to explore many different saccade patterns, or does she tend to continuously exploit a select few patterns, as you would expect. Once habitual behavior is established, the monkey has learned the most efficient patterns for attaining the reward. And here we see a positive relationship between this metric and control energy, suggesting that less entropic saccade selection is less energetically costly. So together, these findings suggest that control energy decreases with increases in efficiency and automated behavior that we associate with habit learning. So those two studies provide some evidence for an association between control energy and cognitive effort. But what about the underlying neurobiology that we associate with cognition and cognitive effort? In order to try and get at that question of whether control energy reflects brain metabolism or glucose utilization in the brain, this study of patients with temporal lobe epilepsy simulated transitions to these 8 different brain states that represent regions that are functionally coupled at rest. So in these simulated states, the functionally coupled regions are set to activity levels of 1, and everything else is set to an activity level of 0. And then after computing the transition energy for these states, that's compared to glucose utilization in the brain to assess whether there's any relationship there. So here you can see this map shows regional hypometabolism in the brains of patients with TLE in the side of the brain that is ipsilateral to the seizure focus relative to the side that is contralateral. And then these regions, or these plots here, show associations between glucose utilization and control energy at the region level. So what's seen across most regions, particularly within the limbic system, is an inverse association between glucose utilization and control energy, suggesting that when glucose uptake is lower, control energy to drive the state transition is higher. And this seems relevant to a sort of energetic inefficiency that is seen in TLE. And then it's also observed that hypometabolism mediates the relationship between loss of volume in TLE and control energy in the hippocampus. Yeah.
In this case, the target path that you're after is simply a transition from that sort of 1s over here and 0s everywhere else to 1s over here and 0s everywhere else.
So it's actually, the initial state is just 0s. And then, yeah, the final state would be 1s in the co-activated one of these 8 patterns and 0 everywhere else.
And what are you using to get your adjacency matrix?
For the structural matrix, or for the control?
It's based on structure still.
Yeah, so the A matrix is still structural. That's from DTI data. Yeah.
And you're free to apply whatever you want, but you're subject to minimizing that cost function you showed us.
Yeah. Yeah, and the glucose data here are from PET imaging. So these findings are consistent with reliance of cognitively effortful processes on brain glucose metabolism. Okay, so just to briefly summarize so far, we've seen evidence that engaging mindful attention, which is a cognitively effortful state, requires more control input into cognitive control associated regions than reacting naturally to cues. We've seen that control energy for transitions decreases over the course of repeated task trials in a habit learning task, and that changes in behavior associated with habit learning are correlated with control energy. And then finally, we see evidence that regional glucose hypometabolism in TLE is associated with greater control energy requirements, reflecting energy and efficiency in the brain. So together, these findings suggest a relationship between control energy, the efficiency of cognition, cognitive effort, and underlying energy demands at the neurobiological level. Yes?
Yeah, I'm just confused by elementary things. So if I go back to your first equation and state in the linear case where the matrices A and B are not functions of time, the transition from a state xt at tan t to a state x at t plus 1 is not only determined by u of t, but it's also determined by the intrinsic dynamics given by the matrix A. And isn't there an energy cost also associated with that transition, even in the absence of a control signal?
So you mean transition in the...
In states. So imagine that u of t is equal to 0. There will still be a dynamical system. X of t plus 1 equals A times x of t. So isn't there an energy cost associated with those transitions?
In the absence of control
Yeah.
input? Yes, I think the control input is what we think of as being necessary to override what would be maybe the natural transition.
Slide where you said this would be the uncontrolled trajectory and this is the controlled trajectory.
Yeah,
I'm wondering whether to adjudicate all the energy expensed only to the controlled trajectory is the correct point of view. Isn't there an energy cost also? Isn't there a confound coming from the energy needed to go through the uncontrolled trajectory? And does your measure of glucose metabolism, for instance, distinguish?
yeah, that's a good question.
I'm just asking this out of total ignorance.
No, it's a great question. Yeah, I think in that case, I guess, yeah, it's important for us to have an uncontrolled comparison to be able to account for that.
Okay.
Yeah, I think I would have to look back in that paper, but I think they also did look at just like random state transitions
Right.
as a point of comparison.
So the comparison would be only due to the control. Yeah.
Exactly, yeah, so then we could get at like what is the input that's just relevant to this specific trajectory. Yeah,
Thank you.
yeah, great question. Yeah.
Maybe you showed it and I missed it. What about the nature of the correlation between areas? So you see that there's some modulation of what happens in particular areas, but if you want to look at in the same subject over the different areas, is there someone accountant doing the math of which area gets what and how they move together, or?
Yeah, that's a good question. So like at an individual subject's level, are the regions correlated?
And is it clear that if you give more to the hippocampus, you have to take away from, I don't know, the brainstem?
I see what you're saying.
I mean, is there kind of a control knob on how do you assign what you have, right? I mean, you need to redistribute what?
Yeah, so in other words, like, is the glucose metabolism sort of regionally redistributed, or?
Or maybe there's a global map of everyone goes to the north, so the south is going to be left.
Yeah, I'm not sure about how the regions compare to one another. That's an interesting question, though. Yeah, like, does there tend to be hypometabolism co-occurring in certain regions, and then do you see any compensation in others? I'm not sure, but that's interesting. Great, okay, so for the last bit, I wanted to switch gears a little bit and talk about some of our ongoing work that aims to extend the linear network control theory to a multi-layer representations of the brain. So we've already talked a little bit about multi-layer networks, but just to recap, they're very useful for modeling systems in which you have components that engage in multiple types of interactions, like in this bird network, for example. These top two layers form a multiplex, or specifically a duplex network, because each individual is connected to itself in each layer. So we might use a model like this to understand how interactions in one layer impact interactions in the other layer. When we're thinking about modeling brain dynamics, multi-layer networks can be very useful for modeling different types of interactions among regions or neurons. We've seen many examples already of where this kind of model could be very useful. For example, if we wanted to model different types of synapses in a single network, maybe different cell types. What we've specifically been thinking about is how to represent both neural activity or functional associations between regions and relations in gene expression between regions. So specifically, we'd have one layer that represents functional connectivity and one that represents correlations in gene expression among regions. So we'd like to understand how we can apply network control theory to drive a multi-layered representation of the brain from one state to another. And this question is particularly relevant to thinking about how the brain transitions between health and disordered states in which maybe disease pathology impacts multiple layers. For instance, we see changes both at the molecular level and at the level of neural activity. And we could then think about how we might target an intervention to affect changes in multiple layers of the system when we can only access one layer. So examples of this might include stimulation-based interventions in which we would inhibit or potentiate neural activity in a particular region of the brain, or maybe pharmaceutical interventions where we might instead modulate activity of a particular neurotransmitter, which would affect changes in gene expression and then ultimately neural activity. So in this schematic, we're looking at a duplex network where we have nodes representing brain regions, between which the interlayer edges connect every node to itself. UT is control input, and we can imagine by administering some control input to one layer of this system, we can sort of drive it along its energy landscape, which would in turn enact a change in the inner target layer with which it makes these directed interlayer connections. So here, as I mentioned, we're interested in modeling functional connectivity and gene co-expression as two layers of a duplex network and determining how we could drive the system from one state to another when we only have access to be able to give input to one of these layers. So in this network, the edges in the functional layer represent correlations in neural activity across time. And the edges in the gene co-expression layer represent correlations across genes between every pair of regions. So then we're asking how much control input or energy is required to push the system from some initial state represented by the origin of the sphere here to a final state 1 unit away. And then what is the associated energy with that transition for each of the two layers. So recently we've started to test this model using data from both mouse and human. I'll focus on the mouse data for today. So to construct a function gene expression network, we're using mesoscale calcium imaging data recorded from the whole cortex. This is data from our collaborator Mike Higley's group at Yale. And this represents about 30 minutes of calcium imaging data collected from mice while they're just hanging out awake on a wheel. So you can see here traces from each of 38 regions of the cortex. And in order to try and capture dynamic changes in functional relations between regions over time, I used a sliding window to compute functional connectivity matrices that each represent about 3 seconds worth of data, or about 30 time points. So each one of these functional matrices then can serve as the function layer in our duplex network. And the gene co-expression layer is constructed from gene expression from the Allen Institute. So I extracted about 7,000 genes that are common between the mouse and human data sets, and then correlated across those genes between every pair of regions to get a co-expression matrix. So we can treat either of these two layers as the input or target layer in our control model.
Sorry, can I ask? So you said you got this from the Allen Institute. Does that mean it's like a resting or like a static snapshot of where you expect to see genes?
Exactly, yeah, from like naive wild type mice at a single point, yeah. So we can then try to address the question into which of these two layers is it more energetically favorable for us to give control input. So here you can see control energy where each one of these points represents one of the functional matrices that I computed, which is paired with the gene expression matrix. And this is energy cost when we are going from function to gene control versus gene to function control. So what you can see is that at some time points, function to gene control is more costly than gene to function. At other time points, it's less costly. And so the next question is, what is different about the network at these time points as compared to these time points? What is it that makes function to gene control more or less costly? So to get at that question, I started to think about what might be different about the state of the mouse across time, since we don't actually have a task or a way to assess differences in cognition. But as I mentioned, the mouse is awake and on a wheel for the duration of the experiment. So our collaborators were able to record 3 behaviors that are generally associated with level of activity and arousal, pupil size, facial movement, and running speed. And if we look at the relationship between control energy at each time point and each of these behaviors, we can see that there's generally a positive relationship between behavior and control energy, such that high levels of activity or arousal are associated with higher energetic cost. And we see this pretty consistently across every mouse. Also related to control energy is the strength of connectivity across the network. So here I just looked at the relationship between control energy and average weighted degree. And we can see that control energy tends to be higher when average weighted degree is higher. And if we look at differences in state, it becomes apparent that there's a positive relationship between arousal and connectivity strength, and then between connectivity strength and control energy, such that higher arousal states seem to be associated generally with higher functional connectivity and higher control input to drive state transitions. Did you have a question?
Yeah. If you go 2 slides back, you talk about this notion of controlling. What is the nature of the control law? Are you saying you are creating something in a 0-1 sort of control law? Is it?
Yeah, so we assume that we're moving one step in any direction. And then we have the matrix represents basically the level of activity or the state. I don't know if that makes sense.
Just trying to figure out what's like, maybe I'm looking at it from a wrong perspective, but if you have a linear control system, a linear dynamic system, there is y is function of the latent state x and then the control law u, right? What is the nature of influencing, what is the nature of u? Is it binary?
Sorry, what is
Is
the
it binary control, like you silence something or you activate something, or is it something graded? How do you cause the transition to happen?
Yeah, so it's a transition from 0 to activity state. Yeah. Let's see. Okay. So we've started to explore the application of multi-layer network control to a gene function duplex. But there are a lot more questions to address, I think, before we could really move forward with this model. In particular, we only have a single static measurement of gene expression at a single time point. So one question is, how could we examine changes in gene expression across time in relation to neural activity? That seems like it would be very important for us to move forward with using this model. And then also, which genes are important for us to include in the gene expression layer? Which genes do we think are important for influencing neural activity? So yeah, question to think about moving forward. And then I also just wanted to note that our group has a preprint recently out that's currently in revision. On using network control theory to study neural dynamics. So if anyone is interested in reading more, or you have a question that might benefit from a control approach, I would definitely recommend checking that out. It goes through the process for determining the inputs to compute control energy or controllability on your data set. And with that, I would just like to thank my advisor, Danny, and members of our lab who contributed, and our collaborators. And thank you all for your attention.